Whether you're a bioprocess engineer hunting for the perfect scaled-down model, a biotech founder architecting the leap to commercialization, or a scientific analyst who craves raw technical truth, you've reached the technical edge. In this space, we live by a single mantra: the process is the product. Welcome to the Biomanufacturing and Fermentation Technology podcast with Prasad Ernala. Hit subscribe and ring the bell to join a global community and stay at the vanguard of biotech translation. Let's build the bioindustrial future together.
Welcome back to the deep dive. We are doing something a little different today. Usually when we talk about strategy, we're looking at, market positioning or high-level decision-making.
The 30,000-foot view.
Exactly. But today, we are going right down to the factory floor. We are talking about biomass separation.
Getting from the fermenter to that initial clarified broth.
I know, for some people, this sounds like a conversation about, plumbing and centrifuges.
It sounds like a maintenance problem.
Right. But looking through the research you've pulled, it's clear that's just the wrong lens. This isn't about cleaning up the soup.
Not at all.
The central premise here is actually quite provocative. The source material argues we need to stop looking at separation as an efficiency maximization exercise. It's not about how fast you spin or how tight you filter. It's fundamentally a risk allocation problem.
That is the crucial shift. If you walk onto a manufacturing floor thinking your job is just to get liquid through a machine as fast as possible, chasing peak throughput.
Which is what everyone does.
And you're actually designing a fragile system. In industrial microbial manufacturing, the goal isn't one record-breaking batch.
No.
It's predictable performance across a whole 6-month campaign. You have to ask, where am I going to put the risk of failure? Because the physics dictates that risk has to go somewhere.
That's a very different mindset from standard process engineering, which often feels like it's about optimizing for the golden batch.
Right, the perfect run.
This approach suggests you're actually designing for where the system breaks.
You're designing the failure mode.
Mm-hmm.
You can allocate your risk to, say, mechanical complexity, like with a high-speed centrifuge.
Okay.
You can allocate it to consumable costs and fouling, like with depth filtration.
Yeah.
Or you can stage the risk across multiple unit operations, but you cannot eliminate it.
So let's unpack that, because robustness is a term that gets thrown around a lot in this industry.
All the time.
The sources break this down into 4 variables that physically bound what's possible, and it seems like the industry fixation is usually on the machine itself.
Always on the hardware.
But the sources argue the first variable is actually the particle population.
It has to be. You can't design a separator if you don't understand the thing you're separating. And we aren't just talking about cell count.
Right.
We're talking about particle size distribution, sure, but more importantly, squishiness or deformability. And then there's the issue of fines.
I want to drill down on fines because this comes up repeatedly as a silent killer in the process. We're talking about submicron cellular debris, correct?
Yes. Tiny fragments of cell wall, pyrrolized material, proteins that have, precipitated out.
And the problem with them is what?
The problem with fines is that they don't behave like particles in the classical sense. They don't settle easily because they have almost no mass, and they don't filter easily because they can wedge themselves right into the pores of a membrane.
Which brings us to the second variable, hydrodynamics. This is where the machine interacts with those particles.
This is where the damage happens. Hydrodynamics is about how fluid moves through the system. Specifically, are you exposing those delicate, squishy biological particles to shear forces? Because if you have a high shear feed zone in your centrifuge, you are effectively taking your intact cells and grinding them into fines.
You're making more of the thing you can't separate.
You are changing the particle population inside the separator.
So the machine is actively making the separation harder as it runs.
In many cases, yes. And that leads to the third variable, fouling kinetics. How does the gunk bust up?
Okay.
Is it forming a cake on top of the filter, which might be manageable? Or is it pore blocking, where the fines get stuck inside the filter structure itself?
And the fourth variable is campaign dynamics. This is the time element.
Right. How does the equipment degradation look over 20 batches? Do the bowls get harder to clean? Do the seals wear, changing the vibration?
But it's more than just wear and tear.
Oh, much more. The most critical part of the time element, and the sources call this the dynamic trap, is that the feed material itself changes over time.
This was the point that really stuck with me. We tend to model the fermenter as a static input. I have 10,000 liters of E. coli broth.
And that's the end of the thought.
But the broth at hour 10 is chemically and physically different from the broth at hour 50.
That is the single biggest blind spot in process design. Let's look at a high cell density bacterial fermentation.
Okay.
In the early exponential phase, the cells are happy, turgid, and intact. They separate beautifully. But if you're pushing for high titers, you're running that fermentation until the cells are stressed, starved.
And when they get stressed, they start to lyse.
They break open. So now, late in the harvest, you have a massive spike in fines, DNA, and soluble impurities. Now, imagine you sized your centrifuge based on pilot data from the early healthy part of the batch.
The machine probably still handles the volume, though, right? The flow rate doesn't necessarily drop.
That's the danger. The centrifuge will meet the flow target. The alarm won't go off.
Okay.
But because the feed has changed, because you have all these fines, the separation efficiency drops. The machine is now passing those invisible impurities right into your clarified broth. It doesn't look like a centrifuge failure.
But downstream, your capture column is about to have a very bad day.
3 days later, your chromatography column fouls, or your sterile filter plugs instantly. And what happens?
Everyone blames the filter.
The plant manager blames the filter. They say, this filter vendor sold us a bad lot. But the filter didn't fail. The separation challenge changed because the harvest timing shifted. You had a particle population problem disguised as a filtration problem.
It's a delayed fuse. You're generating the failure in the harvest, but paying for it in purification.
And that leads directly to the economics. Because if you don't trace that failure back to the physics, you are going to completely miscalculate your costs.
Let's talk about the money. My instinct, and I think the instinct of most people looking at P&Ls, is to focus on CapEx versus OpEx.
The big shiny machine versus the electric bill.
Exactly. How expensive is the stainless steel versus how much electricity or water does it use? But the expert commentary here suggests it's almost negligible.
It's rounding error. In this industry, the dominant cost is almost always lost productive time, downtime. If your plant produces $1 million of product a week, and you lose 2 days because a machine is down or a column is fouled,
That's your entire budget.
it dwarfs the cost of electricity or filter membranes.
But the cause of that downtime seems to vary wildly depending on which technology you picked. It goes back to that risk allocation.
It does. If you choose centrifuges, your downtime is usually shear dominant.
Explain that mechanism. How does shear translate to downtime?
Well, a disc stack centrifuge spins at thousands of Gs. To get high throughput, you often have to spin faster or feed faster.
Right.
That generates shear, particularly at the feed zone where the liquid accelerates. If you damage the product or generate those fines we talked about, you shorten the life of your downstream steps. You might have to stop and clean your chromatography columns every 2 cycles instead of every 5. That is lost time.
So you saved 4 hours on the separation step by spinning fast, but lost 24 hours on the cleaning step later.
Precisely. It's a trade-off. Settling versus shear. Now compare that to depth filtration. If that fails, it becomes consumable dominant, and the failure mode is usually compressible cake formation.
This concept of the compressible cake is really interesting. We're not talking about a rigid barrier like sand.
No, we're dealing with biology. Imagine you're filtering a broth rich in EPS extracellular polymeric substances.
Biological slime.
Basically, biological slime. When that builds up on a filter surface, it forms a gel. As you push more liquid through, the pressure rises.
And that pressure compresses the gel.
Which makes it even tighter. It's a vicious cycle. The cake compresses, the resistance spikes vertically, and suddenly, boom, you're at max pressure.
And the filter's done?
You might have only used 10% of the theoretical capacity of the filter, but you have to throw it away because the hydrodynamics of that cake have shut you down.
So the economic impact there isn't just buying the filter. It's that you have to buy 10 times as many filters as you predicted because they keep choking early.
And then you have to change them out, which takes time. And this links to the other hidden cost, footprint.
Right.
The sources argue that footprint isn't about floor space. It's a proxy for hydrodynamics and cleanability.
I'd love to deepen that, because usually footprint just means, does it fit in the suite?
Think about the geometry. A centrifuge is vertical capacity. It processes huge volumes in a tiny spinning drum. The residence time of the fluid in the pipes is seconds. It's compact.
Okay.
Now look at filtration. It scales by area. If you need to process 10,000 liters, you might need hundreds of square meters of membrane.
That means massive skids, complex manifolds, and lots of piping.
And every meter of that piping is a potential dead leg, a place where fluid can stagnate. If you have a sticky, high solids broth and you have a massive filter skid with 50 valves, how do you guarantee you've cleaned it?
Validation must be a nightmare.
It is. If you can't drain it perfectly, or if the clean in place, the CIP cycle, doesn't reach every nook, you risk carryover between batches. So you spend hours, maybe days, validating cleaning cycles. That is downtime.
I see.
So when you choose a large footprint technology like filtration, you aren't just taking up space, you are taking on a massive cleaning validation burden.
This really paints a picture where there is no perfect machine. Centrifuges shred your cells, filters choke on slime, and are a pain to clean. It feels like you're choosing between two different headaches.
Which is why sophisticated processes move toward hybrid strategies.
Yeah.
This is the risk staging concept. You don't ask one machine to be a hero. You split the job to manage the specific physics of your broth.
Let's walk through the most common combinations. The classic seems to be centrifuge followed by depth filtration.
It's the workhorse. And the logic is, the centrifuge removes the bulk solids, the heavy stuff, using density. The filter then catches the fines and does the polishing using size exclusion and charge.
But based on what we just discussed, you have to be careful with the handoff if the centrifuge is generating fines.
Exactly. If you have a shear-sensitive feed, you accept that the centrifuge creates some debris. You allocate that specific risk to the depth filter. But, and this is a key detail from the sources, you have to monitor the filter correctly.
How so?
Most operators just watch the transmembrane pressure, the TMP, and wait for the alarm.
The sources mention dTMP dt, the rate of pressure rise.
Yes. If you have a compressible cake, looking at the absolute pressure is useless. By the time the pressure is high, the cake has already collapsed. You need to watch the slope.
The rate of change.
If the slope starts to go vertical, you know the crash is coming in 10 minutes. You can switch filters before you fail. That's managing the risk.
What about the double centrifuge approach, using a decanter followed by a disc stack?
That's for when you have high variability. A decanter is a brood. It's a horizontal scroll centrifuge designed for high solids and heavy loads. It's not delicate, but it's tough. A disc stack is a Ferrari high-speed, high-precision, but temperamental.
So the decanter acts as a bodyguard.
The source calls it a variability buffer. If your fermentation is inconsistent, sometimes 10% solids, sometimes 15%, the decanter eats that variance. It produces a consistent, albeit slightly dirty, output. The disc stack then polishes that consistent stream.
So you're paying for two machines to buy process stability.
Exactly.
There was a third strategy that really highlights the disconnect between the lab and the plant. Flocculation. This is adding polymers to clump the cells together so they settle faster.
Flocculation is the siren song of separation. In a beaker on a lab bench, it looks like magic.
I've seen it. It's amazing.
You add the polymer, give it a gentle stir, and suddenly the water is crystal clear and the solids are at the bottom.
But the plant is not a beaker.
No. We call this the hydrodynamic similarity trap. I've seen cases where a process worked perfectly in the lab. They designed the whole plant around it. But in the plant, the flocculated material had to travel through 50 meters of pipe, 3 90-degree elbows, and a control valve to get to the centrifuge.
And that turbulence just ripped the flocks apart.
By the time they hit the separation zone, the beautiful large clumps were gone. They were back to being fine dust.
Oh, no.
The chemistry was right, but the hydrodynamics were wrong. They wasted millions on a system that couldn't work because they ignored the shear in the pipes.
That's a brutal lesson. It's not just about the separation unit. It's about the journey the fluid takes to get there.
The pipe is part of the process.
I want to shift to the invisible risks. We touched on the idea that separation failures rarely look like separation failures. This has huge implications for GMP good manufacturing practice and quality control.
It does. If a seal on a centrifuge starts to fail, or if you have a slight drift in cleaning efficiency, you usually don't get a flashing red light that says contamination.
You get a bioburden spike 3 days later.
Or you get unexplained yield loss. Let's say your centrifuge performance drifts slightly. Maybe the discharge isn't quite as clean. You are carrying over more host cell protein, or HTP, or DNA.
Okay.
That extra gunk binds to your product or fouls the binding sites on your expensive chromatography resin.
So your yield drops by 10%.
And the investigation team tears apart the chromatography column. They blame the resin. But the root cause was a subtle hydrodynamic shift in the centrifuge 3 days prior.
And nobody connects the dots.
The causal chain is centrifuge drift, right arrow increased DNA load, right arrow downstream, fouling, right arrow yield loss. If you aren't monitoring the separation physics, you will never find the root cause.
This brings us to single-use systems. We see this massive trend toward disposable plastics bags, tubing, capsule filters. The argument is usually about speed and eliminating cleaning. But does that solve the risk problem?
It solves the cleaning risk. You don't have to validate CIP for a plastic bag, you just incinerate it. So cross-contamination risk drops near zero. But remember, you cannot eliminate risk.
You can only reallocate it.
You reallocate it.
Where does it go?
It goes to the supply chain and to E&L extractables and leachables. You're now relying on your vendor's quality control. Did they cure the plastic correctly? Are there chemical additives leaching into your drug product?
And I imagine the logistics are not trivial.
It's a warehouse problem. If you're running a large process, the volume of plastic you need is staggering. You're trading a steam pipe for a warehouse full of palletized plastic bags. And if the supply chain breaks,
Your plant stops.
your plant stops. You've traded an engineering risk for logistics risk.
Finally, I want to touch on AI and digital twins. Everyone wants to put AI on their bioprocess these days, but the source material is pretty critical of the throw data at it approach.
The critique is valid. The rule they propose is use mechanistically meaningful features.
Which means
It means don't just dump raw sensor data temperature, inlet pressure, pH into a black box algorithm and hope it predicts a failure. Those are just numbers. You need to engineer features that represent the physics we just discussed.
give me an example of a mechanistically meaningful feature.
Instead of feeding the AI the pressure, feed it the calculated resistance of the cake, or feed it the slope of the turbidity change over time.
So you're teaching the AI to look for the compressible cake or the shear drift.
Exactly. If the AI understands the mechanism, if it knows that a rising slope in resistance means the cake is compressing, it can predict a blockage hours in advance. If it's just looking at raw pressure, it might not catch it until the alarm trips.
You have to embed the physics into the code.
You have to.
This has been a massive reframe. We started this by saying separation isn't about plumbing, and I think we've proven that. It's about risk, physics, and economics all tied together. For someone listening, maybe a process engineer or a plant manager, what is the one thing they should change about how they walk their floor tomorrow?
Stop looking at the unit operations in isolation. Don't look at the centrifuge and ask, is it spinning? Look at the broth entering it and ask, how is the physics of this fluid changing?
What's a hidden variable?
What's the hidden variable? Is it the fines? Is it the compressibility? Is it the variability?
And once you spot the killer variable,
You ensure your equipment is designed to buffer that specific risk. Don't try to make one machine do everything perfectly. Stage the risk so that when the harvest quality drops, and it will, the system bends instead of breaks.
it's about building a system that can handle the bad days, not just the good days.
The good days are easy. Anyone can separate a perfect broth. You get paid for handling the mess.
I think that's the lesson. The fermenter to broth step is the gatekeeper. If you get the risk allocation wrong here, you pay for it in every single step that follows.
Absolutely. And I'll leave you with one final thought, perhaps a bit of a warning. We often see data from lab scale, little 50 milliliter spin tube or a small filter disc, and we think, great, the separation works.
It's the it worked in the Beaker fallacy.
A robust separation at lab scale is often meaningless. If you don't account for the flow distribution of the big manifolds or the shear differences in the industrial pumps, you aren't designing a process. You're just gambling on hydrodynamics.
You're just hoping.
You're hoping the physics at 10,000 liters behaves like the physics at 10 milliliters.
And as we've learned today, gravity and shear don't scale linearly.
They certainly do not.
A sobering thought, but a valuable one. Thank you for breaking this down. It's definitely changed how I'm going to look at those stainless steel tanks next time I see one.
My pleasure.
And thank you for listening. Next time you're troubleshooting a process, ask yourself, am I solving a separation problem, or am I just seeing the symptoms of a risk I didn't allocate? See you in the next deep dive.
Hi there, thank you for staying with me until the very end. If these technical insights help bridge the gap in your own process, please hit subscribe and make sure to hit the notification bell. Stay connected for our next deep dive into the realities of biomanufacturing. I am Prasad Ernala. Thanks for listening.